स्टूडेंट्स को अनम्यूट मैंने करना है या खुद करके अनम्यूटेड है ये माइक सिस्टम वैसे इसके अंदर है इनको ये एक्चुअली चाहिए Assalamu alaikum everyone. Let me first let me first introduce myself. I'm Dr. Rubeena. I've just joined CPMC about 15 days back, and this is my first lecture and first interaction with all of you, and also the first interaction with the subject of microbiology. So I hope we have a good rapport between us, and whatever I teach you, you understand. And basically, I would like all of you to put up any questions if you think that I am going too fast, or if there is something which you do not understand. Microbiology constitutes about 60% of your subject, which is going to be assessed in the final examination of yours. So 60% weightage has been given to microbiology, and another 40% is given to general pathology. So it's a huge chunk of your curriculum which you need to study. And microbiology is not, it's an easy subject, but at the same time, it's a bit difficult to remember. So you need to go through it over and over again, and build up your concepts and build up the basic knowledge about microbiology, on which you can then build up on the different subjects. So let's go ahead with the first lecture, which is about the introduction to microbiology. And today I will give you we start with a basic overview of the subject. What is important in microbiology? Why do we need to study this subject, and how it is going to impact our different clinical scenarios which we are going to come across. So what are the teaching objectives of today's lecture? Basically, to give you an overview and to introduce you to the subject. Why is it important? What is its definition? What's the differences between a pathogen and a commensal? And what are the main organisms which we are going to study, and how do they differ from each other? This is the book which is followed by all, and this is the book which has been recommended by the UHS as well. It's an easy book to follow, but at the same time, it's I find that it is a little bit difficult to retain. So for that purpose, I always suggest to my students that they should take up some sticky notes, and on any page where you are studying, you just need to put down the points in a bullet form. For example, this is the chapter on cell walls of, this is a page which is giving you the cell walls of gram-positive and gram-negative bacteria. Yes, Amna?
मैम आपकी आवाज नहीं आ रही, लाइक बहुत ज्यादा ब्रेक हो रही है.
आवाज नहीं आ रही. अच्छा, I'll look into it.
Hello.
Okay. Now can you hear me? Is it better?
Yes, ma'am. Yes, ma'am.
Okay, let's go ahead. The cell walls of gram-positive and gram-negative bacteria, as I was saying, this is which is being reflected on this page. So it's a huge chunk. And if you just write down the bullet points of what is in this paragraph, it would be very easy for you to remember and recap when you are going through a revision of the topic. So this is one way which I suggest for you to make your small notes as you go along each chapter. Make the book your own. By this, I mean that you can use different ways to understand and to learn this book. Make sticky notes, as I told you already, or something which is very, very important are the diagrams in this book. At times, the diagrams are going to give you a whole paragraph in a very brief visualized form. So what you need to do is to learn to draw these diagrams and to add those points which are not mentioned in the paragraph to that diagram, so that it becomes a composite, which when answering a question, you can just remember that diagram instead of going through a whole page of the subject matter which is being given. So at times I find it very easy to remember a diagram, jotting down the salient points which are mentioned in the paragraph, and then utilizing them for answering that subject. Make flow charts. This is a very easy method of remembering the organisms and their characteristics. As you go along the book and we study different organisms, you will realize how easy this flow chart methodology makes it for you to remember the different points. And then make use of the bullets, as I already told you. If it works for you, maybe all these things work for you, maybe only a few of them work, or just one, or none of them. It is your own it's your own aptitude which is going to make you understand this book and how you follow this book. These are just suggestions. So what does microbiology deal with? Basically, it's the science of the organisms or the microbes which are present around us and within us. So they are present all over the world in the biosphere, even on our bodies, within our system, and you would be surprised to know that there are more bacterial cells in our human body as compared to the human cells. And most of them, they are in those areas which are in contact with the exterior. So it becomes easier for these organisms to reach the person either on the surface or through the GIT into the intestines, into the mouth. So there are various areas where these microbes are going to be found even within our systems. So the question arises, what are the different types of microbes? How do they differ from the animal and plant cells? Where do we find them? Where do they live? How do we classify them? What are their beneficial effects? Do we get any benefit from these humans and animals and plants? Do these have any harmful effects? How are they going to affect our body system? How are they going to affect the environment around us? Just to highlight the importance of infectious diseases. In 2020, we had COVID-19. Before that, we have another number of infectious diseases occurring. But one thing which was consistent with all of these was that the infectious diseases have ranked either third or fourth amongst the leading causes of death, not only in the US, but all over the world. Heart disease and different types of cancer, they rank 1 and 2, and after that comes the infectious diseases. So you can understand the importance of these organisms and the diseases which they produce. So what is microbiology? If we split up this word, we can split it into three parts. Micro, meaning something which is too small to be seen by the naked eye. Bio means life, and ology means study. So anything which is too small to be seen by the naked eye, excuse me, and it is impacting our life, it is livable, and we are studying it, that is what is microbiology. So basically, it is the science where we are studying small living organisms which are not visible to the naked eye, and they are called microbes. We can call them organisms, microorganisms. Mostly, we can call them pathogens when they are disease producing, and the layman term we can also call them as bugs. So what is the importance of medical microbiology? There is microbiology which is going to deal with all types of organisms producing a disease or beneficial or harmful in the environment, in the plants, in the animals. But when we are talking about medical microbiology, we are restricting ourselves to those organisms which are the causative agent of human infectious diseases. And what are the changes which are they going to be producing in our system? It is going to deal with the etiology, which is the cause, pathogenesis, how does the organism produce the disease, lab diagnosis, how do we identify the toxin, how do we identify the cause of that infectious disease, then what is the specific treatment, and how do we control them, basically the vaccination or the immunization. So we have microbiology, medical microbiology dealing with the infection, the infective dose, the colonization, transmission, complications. All of these will come under the heading of the medical microbiology scenario. And basically, it's concerned with two types of organisms when we are talking about the human system, pathogens and commensals. Anybody have an idea what is a pathogen? Anyone? Can any?
Pathogen
Yes.
Which can
can be
cause
both
infection
bacteria
or
and fungus or any other virus thing which can produce harmful effects to the body, any outside or microorganism.
Okay. Yes, I could hear another voice as well.
It's any organism that causes disease.
Okay. Any organism that causes disease. Yes, good. So what is a commensal then?
No idea, ma'am.
No idea. Okay. Rida, can you give me an answer?
Ma'am, could you repeat the question or ask
What
it?
is the commensal?
I don't know, ma'am.
Okay. So let's go ahead and we come to the definitions of pathogen and commensals and what's the difference between them. So basically, this is a microbial world which we are going to talk about, and it is going to be constituted by bacteria, viruses, fungi, and parasites. And when we study them individually, they are going to be known as, if we are studying the bacteria, it would be bacteriology. If it's viruses, virology, parasites, parasitology, and fungi are the mycology. You may have heard all of these terms before. And all of these, they are trying to harm us, whereas our immune system is trying to prevent the infection in our body. So as long as our immune system is working, we are strong and our immune system is strong, we are able to ward off all of these types of organisms and remain healthy. These are the infectious microbes which are present around us and which are producing different types of diseases in individuals. It could be a bacteria, it could be a virus, it could be a fungus, it could be a parasite. They have different morphologies, they have different shapes, they have different sizes, and different characteristics which put them into different classes. Coming to the term pathogen and commensal. Pathogen, as rightly said by Zan and Rida, it's a disease-causing organism. But a commensal is also an organism which is non-pathogenic. That is, it is not going to produce a disease. So, why are we concerned with a pathogen and a commensal? Why are we concerned with a commensal? Pathogen, I can understand. It's a disease-causing organism. So all of us are interested in knowing what it does. But why a commensal? Because certain...
Ma'am, commensal is like living in a relationship.
Yes.
One can get a benefit and
Symbiotic
other may be
relationship.
harmful.
Yes, exactly. Commensal, they are present in our bodies as well. They've got two functions mainly. Number one is that when they are in our body, they are going to prevent the pathogens from taking a hold. For example, if you have a society or an area in which there are 10 houses and there are 10 people living in those houses, can an outsider come and occupy any of those houses?
No, ma'am.
No. Similarly, if in our system, in our intestines, we have these commensals, they are present over there. So if they are they have been occupying the intestinal walls and they are taking the spaces over there. So it is very unlikely that a pathogen will find a place to come and reside over there. Secondly, since the commensals are using all the food and all the material which is present in that environment, a pathogen is not going to be likely find the nutrition which it is needing to survive. So in the presence of a commensal, a pathogen is unlikely to find a place to settle down, to multiply and to produce a disease. It's only in those situations when we have a decreased number of commensals that the pathogen is going to take over and produce a disease. Secondly, at times, especially in immunocompromised individuals, in those individuals who have some disease, for example, a patient who's diabetic, a patient who has some immune problem, a patient who has cancer, it is those individuals in whom these commensals, they become very important because since the immunity of that individual is lost or it is decreased, it is these commensals which can multiply and start producing a disease as well. So these are the two important reasons why we need to know the areas where a commensal is present. And if we are looking at that area due to some infection or some other problem, then we need to consider not only a pathogen at that site, but also as a commensal, keeping in mind the individual's immune status. Clear?
Yes,
Yes,
ma'am.
ma'am.
So what does a pathogen have to do to produce a disease? It has to, number one, infect or infest. Can anybody give me the difference between infect and infest? Anyone?
Ma'am, entrance.
Yes.
Ma'am, infestation is basically crowding the place, and infecting is like really damaging or infecting the other person.
Okay. Anybody else? Anyone
Ma'am,
else?
infestation means किसी को affect करना in a large amount.
No. Infect and the difference between an infection and an
Ma'am, infestation.
Yes.
Infestation में parasites होते हैं और infection में bacteria या कुछ ऐसा.
Shabash. But that is correct. At the same time, beta, there is a difference, a little bit more of a difference. Infection is basically when an organism is producing a disease inside your body. When an organism is able to go inside and produce any sort of a disease, that is an infection. When it stays on your skin surface, it doesn't go inside. That is an infestation. And a common example of this is infestation is lice in the hair. Understood? It's just on the surface. It's not going inside. It's not harming the interior of your body. So that is referred to as an infestation. But infection, even on the skin, would be resulting in some sort of a problem. For example, we have boils, we have acne on a surface, on a face. All these things, they are not an infestation. They are an infection because the bacteria or the organism has broken the skin and produced that pus and produced that lesion. In infestation, there is no breakage or breach of the skin surfaces. Clear?
So, ma'am, basically we can say infection is when an organism goes inside our body and it does damage to the inside of the body.
Yes. Yes.
And an infestation is basically an organism producing harmful effects on the outside of the body.
Definitely, yes, correct. Number one, pathogen needs to infect a host. Once it infects the host, then it needs to reproduce or replicate itself. Because if it is just present in a few numbers, it is not liable to produce the damage, or the body system is going to overcome it and it is going to be destroyed. So it has to reproduce in numbers so that it can spread, it can cause damage, and then ultimately the infection will result. And finally, it needs to ensure that the progeny it is producing that can also be transmitted to another host. So if an infection is occurring, we can take the example of COVID. If the COVID stayed in one individual, this mass problem would not have occurred, but it is being transmitted from one person to the other. There are different ways of transmission. It could be the airborne, as we are experiencing with COVID, or it could be a contact as occurs with measles. So there are different ways of transmission, but the organism needs to be transmitted from one person to another to continue this chain of infection. Yes, Anna?
Ma'am, this wouldn't apply to non-contagious diseases. Contagious diseases may it ensures its progeny and transmission.
Yes.
So what would we say about non-contagious diseases?
Certain non-contagious diseases may occur. Just say, for example, we can take the example of salmonella. अब salmonella में आपकी जो organism है that is being passed out into the stool. Although there is no direct human-to-human contact, but the organisms, they are being passed out into the fecal matter, and through our improper hand washing, improper hygiene, they can be transmitted to an individual who's taking food, who's coming into contact with food, and then it is going to be transmitted to the other individuals. So in that respect, it's not a contagious disease, but the progeny is still being transmitted to another. This chain of infection will not be there if it will not survive if you break any link out of it. So if you break the transmission, you are able to contain the infection. And that is basically how infectious diseases, they are being controlled all over the world. You break the chain of infection at any level. Either you stop the source of the infection, or you stop the transmission, you stop the replication. Whatever aspect you stop, that is going to break the chain. So progeny, if you stop it at the point where the reproduction is stopped, then naturally the progeny will not be transmitted. If you transmit, if the organism is being transmitted through the aerosols, then if you stop that, that is the reason why we are all wearing masks. If you stop that, then you are again preventing the transmission. So until and unless the pathogen infects, reproduces, and transmits, it will die off. Its viability depends on all these factors.
All right, thank you, ma'am.
So why are they important? Why do we need to study all these infections? They have played an immense role in history. They have caused a lot of death, lost a lot of misery, and at the same time, they are causing a lot of anxiety, as we can see with COVID-19. How many stressful conditions people are going through, a lot of stressful conditions because of this. And most importantly, we are coming up against antibiotic resistance, which is a huge factor. Giving you an example of, this is an example of the Spanish flu, which occurred in 1980. Wards and wards of people, they were filled with people, and they had the Spanish flu, and then it went on for a couple of years, and then it died off. Similar to COVID, which is, we are in the second year of COVID. And then this picture, can anybody tell me why I put up this picture over here?
TB?
TB. Because if tuberculosis had not been present, maybe we would have had him with us for a longer time period, and maybe Pakistan's history would have been a bit different. Okay. So, and every day we get something in the news as well. Currently, it is COVID. Before that, it was SARS. Then it was hepatitis. Something or the other is always keeping these infectious organisms in the news, and they are also benefiting us in different ways. They can help us by making the food, the medicines, and they also help the ecosystem in decomposing and in recycling the nutrients which are present in the environment. At the same time, they have harmful effects, and these harmful effects could be in the form of contamination of the things around us, an exposure to the individual, exposure to the animals. Consequently, these animals come into contact with us, and ultimately, there is some active disease. Similarly, they can also contaminate our food, our water, our blood. So there are different ways in which these organisms can benefit as well as harm us. Coming to the two important domains when we are going to study the eukaryotes and the prokaryotes. Amongst the eukaryotes, we have the animal cells or the human cells, plants, fungi, and protists. Whereas in the prokaryotes, the most important which we are going to be concerned with are the bacteria. The basic structure of a prokaryotic cell and the eukaryotic cell, there are certain differences amongst the two. Eukaryotic cell is a human cell. We'll take that as an example. You've gone through a normal human cell. Today, we are going to concentrate on the prokaryotic cell and how this is different from the other types of organisms which we are going to deal with. So, when we talk about prokaryotic and eukaryotic cells, these tables are important to remember because they are going to form the basis of starting your viva. They are going to give you the salient features which are going to help you in differentiating between a human cell as well as a bacterial cell. There is a nuclear membrane in case of the human cell, and the cell has chromosomes more than one, and it undergoes mitotic division. In case of a bacteria, it's a very simple cell. It has a chromosome, but it is just a one in number, and it's a long thread which is just wound up in the bacterial cell, and there is no nuclear membrane, and it does not undergo mitotic division, rather it undergoes binary fission. Then the mitochondria, they are going to be present in the eukaryotic cells, whereas we just find a few organelles in the prokaryotic cell. The size of the ribosome is 70S in prokaryotic bacterial cells and 80. And this becomes a huge difference which helps us in antibiotic targeting. So when we make an antibiotic, and if you want to target the ribosome, that makes it very specific for the bacterial cell because the human cells will be spared due to their size of the ribosome. And then there is a difference in the bacterial cell. Why does the size matter? Can anybody give me why I've put up this picture over here? What do you understand by this picture?
It is easy for the virus to cross certain sites.
Okay. What else? Just to show you that this is a normal size of it. This is the approximate size of the human cell. And look at the size of the prokaryotic cell, which is the bacteria and the virus. So it is very easy for these organisms to go inside these cells and to destroy them. Viruses, they cannot multiply outside the cell. Bacteria, they will multiply outside the cell as well as within the cell. So our human cells are larger as compared to the bacterial or the viruses, which makes it easier for these organisms to get inside and to destroy the machinery of the human cell. So different types of organisms which we are going to study. Number one is the bacterial cell, and these are the various structures which you can see. We'll go into the detail later on, but simply this is the interior which is made up of the cytoplasm, and the chromosome is just lying there as a jumbled mass, and it's a single thread loop which is going to be present within the cytoplasm. And the most important feature of the bacterial cell is its cell wall. And then it has various organisms which are going to, structures which are going to help in the movement, in the adherence to the cell surfaces, and in making it viable within the human environment. This is the example of a virus. There are different types. Like you can see, there's a naked virus. Naked virus means that it does not have a capsule or a covering around it. And this is an enveloped virus over which you can see the capsule or the envelope around it. So this becomes more protected, this is less protected, but they are as lethal whether they are naked or they are enveloped. And then we have different other categories like the bacteriophage. This is basically a virus which infects a bacteria. And we, as we study virology, will study these different organisms. Now, again, this is a chart which you need to remember, and this is going to help you in attempting not only your SCQs, but also your MCQs and your viva. Because generally, differences between viruses, bacteria, fungi, and parasites, they are going to be asked from you often. And the most important differences which are going to be asked are between a virus and a bacteria. As I showed you the diagram, viruses, they do not have a cell wall, and they are smaller as compared to a bacteria. Nucleic acid, in case of a virus, which differentiates it from not only the bacteria, but also the fungi and the parasites, is that it contains only one type of nucleic acid. It will either be a DNA or an RNA, never both. Whereas in case of the bacteria, it is the DNA as well as the RNA which is going to be present within it. And the virus is not a typical cell. It does not have a nucleus. It does not have the ribosomes or the mitochondria. And it is not motile. It does not multiply through binary fission or mitosis. So all of these characteristics makes it a simple organism which is unable to survive on its own if it does not go into a cell. So a bacteria is able to survive in the extra as well as the intracellular environment. Whereas on the other hand, the virus needs a cell for its multiplication. Since it does not have mitochondria, it does not have ribosomes, how is it going to replicate the DNA? How is it going to replicate its RNA or DNA? And how is it going to produce the various proteins which it requires? So what happens is that the virus goes into a cell, it disintegrates into different structures, its proteins, its RNA, DNA, and then it utilizes the machinery of the host cell to make up all these structures. And then they get reassembled and the virus is then thrown out of the human cell. So that is how it survives. Bacteria, it can survive without all these things because it can survive without the human cell because it has got all the components, as you can see. It has the DNA, RNA, it has the ribosomes, it has certain, it divides by binary fission, it has certain organelles which helps in its multiplication and in its metabolism. So these are the basic differences between these two organisms. Now, we come to the gram stain. This gram staining is going to be a very, very important learning point for you because this is the basis of the whole of microbiology. You will do in your practicals when you inshallah come back, you will be doing the gram stain procedure. But you need to remember that this is what makes microbiology what it is. It helped the Danish physician Hans Christian Gram into dividing all of these organisms which we deal with in medical microbiology or elsewhere into two different groups. And what he did was he experimented with a staining procedure which was able to stain these organisms and put them into two categories. And the stain utilizes, which is number one is the crystal violet, which this is a glass slide, and you have these bacteria on it in the form of a smear. And when we put the first stain, which is the crystal violet, all these bacteria, they are going to take up the...
Ma'am?
Yes.
Ma'am? Ma'am, आपकी आवाज नहीं आ रही सही से.
अच्छा. अब आ रही है?
जी, अब आ रही है.
Okay. When we start with the gram staining procedure, what we do is that we take a slide, whatever bacteria we want to stain, we just make a smear. Smear is just, what can I call it? All of you know what a smear is? It's just...
Yes, ma'am.
Yes, ma'am.
Yes, you make a smear and then you are going to add the first stain, which is the crystal violet. It's a purple colored stain. When we put it onto that smear, all the bacteria which are going to be present in it, they are going to take up the purple stain. The second stain we add is the grams iodine. Grams iodine acts as a mordant. Anybody know what's the definition of a mordant? It's an English word. Okay, mordant basically is any chemical which is going to make the other chemical stick to it very strongly. So I can give you an example of yes, dyers. If you see a dyer, when he's got that whole big vessel in which he has the water and he has that colored dye in it, and then he dips the cloth in it to put it into a particular color, he is also adding salt to it. And salt acts as a mordant, allowing the color to get attached to the cloth. Similarly, and jisko kehte hain na rang ko pakka karna, salt acts as that. Similarly, over here, the Graham's iodine is binding the crystal violet to the bacterial cell wall and not letting it go out. Now, the difference comes here with the addition of acetone. Acetone will decolorize all those bacteria in which the Graham's and crystal violet complex is not formed. So there is a structure of certain bacteria which will allow the crystal violet to penetrate and which will allow the Graham's iodine to form a complex with crystal violet. And this complex is then big enough to not come out of the cell wall. But another group of bacteria in which this is not happening, the Graham's iodine in crystal violet, they do not remain inside and they come out and they are then decolorized with the acetone. So after decolorization with the acetone, we add a counter stain. The counter stain is, for example, methyl red or safranin. And these are the dyes which are going to be lipid, they can pass through the lipid layers. So all those organisms which have more lipid content in their cell wall, they will take up the counter stain and they will start appearing pink. So if you are performing a Graham stain and you have a mixed colonies of bacteria, some of them are going to stain purple and some of them are going to stain pink. So whichever stains purple, we say that it is Gram positive, and whichever stains red, we say it is Gram negative. So with red negative and with the purple, it is with the P, it is positive. And hence he was able to put the whole of the bacterial organisms into two groups, Gram positive and Gram negative, and this was based on the differences in the cell wall of these two bacteria. So when we see them, the Gram positive would appear, and then he was also able to visualize the shapes because he could now see the bacteria. He could also see what types of shapes they have. So there were two groups, Gram positive and Gram negative, and then they were further subdivided into Cocci and Bacilli. These were the two broad groups into which the whole of the bacterial population could be divided. So then, as I told you before, the differences are based on the differences of the Gram positive cell wall and the Gram negative cell wall. Just briefly over here, we'll discuss in detail later. A Gram positive cell wall has more peptidoglycan in it, and peptidoglycan is a very thick layer which has meshes in it or spaces in it, which allows the Gram iodine and the crystal violet to go inside, form complexes, and then these complexes are big enough that they cannot get out. And so the cell wall appears purple. In case of Gram negative, we have very little peptidoglycan, but we have a lot of lipid. So the dye, counter dye, that is the safranin, it can go inside and then it takes this, it gets bound to the lipid and it does not come out again. So these organisms, they will appear Gram negative. So a more closer view of the Gram positive and the Gram negative organisms. Now, when we are seeing the Gram stained picture, we are then also seeing the shapes of the organisms. Basically, there are three different shapes: Cocci, Bacilli, and the spiral-shaped bacteria. Majority of the organisms are either the Cocci or the Bacilli, and very few fall into the third category. And reproduction, which is responsible for the different shapes of the different arrangements of the bacteria, is basically a binary fission. This is a bacteria and it is just going to divide into two, and one single parent cell is going to give rise into two daughter cells. This picture just reflects the division of budding, and this happens in case of the fungi. how does the particular arrangement of a cell occur? This is because of the division in different planes. In this plane, you have, in this diagram, you have a coccus in which the division is occurring in the vertical plane. And when they are divided fully, then they are going to go towards the side and separate from each other. If the two remain stuck to each other, then this organism is going to be called a Diplococcus. Diplo from two and the coccus because of its shape. So this is a Diplococcus. And then if these keep on dividing, this again divides into two, and this divides into two, and this becomes four, and they remain attached with each other. That is how these chains are going to be produced, and these are known as streptococci. Why are we so concerned about the division and about the arrangement of these organisms? Because this becomes an important characteristic apart from the gram staining reaction of the bacteria, which will help us in identifying a particular pathogen. Similarly, over here, you can see that it is dividing in two planes at right angles to each other. And this is going to result in a tetrad. And this morphology will help us in identifying the micrococci. So basically, the arrangement is going to help in the identification of the bacteria. So the same thing, this over here is going to result in chains. This is resulting in diplococci formation. This is a tetrad formation. And when you are having divisions in different types of planes, it's not just rectangle or at right angles to each other, or it is just not in one plane, then you get a haphazard division of the bacteria, and that results in the formation of clusters. And this is the cluster formation. This commonly known as the Staphylococcus, and this is commonly known as the Streptococcus, and these are the two major bacteria which are going to be very, very important in producing disease and from your curriculum point of view. This is what we see under the microscope after gram staining of these organisms. These are the chains, these are the tetrads, and these are the clusters. Same principle applies to the bacilli or the rods. Sorry, the spelling of bacilli should be double L-I. The bacillus single, and again, if it goes into this vertical division, it will form these chains. If this division is in this plane, then it will form these palisades, which are side by side, standing with each other. And if these two separate, and these are then known as diplobacilli. And this is what you will see in the microscope. These are gram-negative gram-negative bacilli. Again, over here, you can see the curved organisms. Over here, you can see the gram-positive organisms. Over here, again, this is the arrangement. This is still not completely divided, and it's going to separate from this B, and then it will occur. It will become 2 in number. Then there are certain rods which are spore formers. What are spores? Spores, basically, these are giving the vegetative cell the edge of survival under abnormal conditions or under the conditions in which they are not able to survive. For example, if the vegetative cell needs moisture, it needs temperature for its growth and multiplication, and the environment is very dry and the environment does not have, and it has high temperatures, some of these organisms, they are able to convert themselves through a cycle into a spore, which has a very thick outer layer, and this thick outer layer is then able to it is able to survive the high temperatures. It is able to survive the dry conditions, and it can survive for many, many years in the soil or in the environment. And consequently, it is going to enable that bacteria to live for many years. And whenever the pH, the temperature, and the outside conditions are conducive, this spore will generate or germinate, and it will go back into the vegetative cell formation. So if the circumstances are good, the spores can go back into the vegetative cell. If the vegetative cell finds the circumstances are poor, it forms a spore. And this is the spore which we can see within a gram stain picture or through special stain. And these also become one of the characteristics by means of which we can identify these organisms. These are the spiral forms. They can be seen in a blood film. They can be seen in different tissue films, and they indicate the presence of spirochetes. Then there's certain different bacteria. Again, they can be identified on the basis of their morphology, their arrangement, and what we can see under the gram staining. And these we will study one by one in a special bacteriology. Just to give you an overview, how we classify these organisms or put them into different categories. Now, first of all, we have the gram-positive cocci. And if they are in clusters, then we call them as Staphylococci. And then they are subdivided into different species based on their different biochemical reactions. If they are in chains, then we call them Streptococci. Again, there are different species of Streptococci, and they can be identified with different biochemical tests or different different types of antibiotic sensitivity testing. Then we have the gram-negative organisms or the gram-negative cocci. Amongst the most important is the Neisseria and Neisseria meningitidis, which is the causative agent of meningitis. And this is the gram staining. In gram-negative rods, we have a long list like Salmonella, causative agent of typhoid fever. We have Vibrio, causative agent of cholera, Shigella, different types of organisms, and these are basically going to produce a disease in the intestine of individuals. Then there are certain gram-negative rods which are very difficult to grow. So, and they produce certain different types of diseases. They are not within the GIT, they can be anywhere in the body. For example, Haemophilus influenza causes influenza, Bordetella, it causes whooping cough. And for this, the children are given a vaccination in early childhood. Then we have the gram-negative rods, which are anaerobic. That is, they require an environment which does not contain any oxygen. And these are again present in our bowels, in our intestines. Gram-positive rods, Clostridia, again, these are anaerobic organisms. We have Bacillus cereus, which is a causative agent of food poisoning, and this is an aerobic organism. So these are the different categories of organisms which are important, which we are going to study, but you need to remember them when you, in the upcoming lectures, you will be given a lecture on classification of bacteria. And then you need to remember these organisms because sometimes, not sometimes, but rather a very important Viva question. Your Viva starts with name the gram-positive organisms. And as soon as you say Staphylococcus or Streptococcus, then the examiner starts asking you about Streptococci or how do you differentiate between Staphylococcus and Streptococcus. So until and unless you know which category these organisms fall into, you will not be able to continue with your Viva effectively. Then there are certain bacteria which do not take up the gram stain. And that is because of their different characteristics which they have. For example, Mycoplasma, they do not have a cell wall. Mycobacteria, they have mycolic acid in the cell wall. This is a picture of Mycobacterium tuberculosis, which causes TB. And so because they have different structures of the cell wall, which is different from the normal bacteria, they will not take up the gram stain because gram stain is being targeted for certain structures in a bacterial cell. Similarly, spiral bacteria, they cannot be stained. They are seen either with specific stains like the silver stain, or you use specific microscopes for their visualization. Then we are going to study certain bacteria which are intracellular. And since they are intracellular, gram stain will not reach within the cells, and so they cannot be seen. They do not survive outside the cell, so we are not able to see them. For this purpose, we require either immunofluorescent microscopes or some other staining which is going to visualize them in the tissues. Till this point, any questions, Beta? Anything which you fail to understand? All good?
Yes, ma'am.
Okay, let's go ahead.
Yes, ma'am.
Then we are going to study the structure of the bacterial cells. After this, this is going to be today and tomorrow, and this is going to help us in understanding the morphology of the bacteria, including the spores, the morphological basis of classification of bacteria, how the cell wall is important in putting them into gram-positive and gram-negative cell walls, and how does they contribute to the antibiotic effectiveness how does the cell wall difference is responsible for the antibiotics to be effective against a particular bacteria? What are the different structures of a bacterial cell and what are their functions? So we've discussed about the eukaryotic cell types and the prokaryotic cells, discussed the size, we've discussed the two types of cells and the differences between them. One important thing I need to emphasize over here is the surface area. The surface area of the eukaryotes is much greater as compared to that of the bacteria. Despite that, the eukaryotes need structures and organelles for their survival and for their metabolism and for their multiplication. Whereas in case of eukaryotes, the surface area to volume is 3:1. So the surface area is such huge that in a smaller volume, the bacteria are able to take up more nutrition through that greater area, and it reaches all the parts of the bacterial cell, and therefore it is able to utilize it for its energy and for its metabolism. Differences between the prokaryotic and the eukaryotic cell, we've already discussed them, again highlighting the importance of your knowing these differences. Coming to the bacterial morphology. Coccus, then we have the arrangement of a coccobacillus, which is in between a coccus and a bacillus. Then there are curved organisms like the vibrio and the spiral organisms like the spirochetes. Chains we discussed was a streptococcus, cluster was a staphylococcus. Similarly, a bacillus and a vibrio, spirillum, spirochetes. And these are the different bacterial shapes and arrangements which we've already gone through, and you need to sort of know how to draw these various shapes. Binary fission, just dividing each into two, and this is the arrangements which we went through. This is just a recap of what we've already done so that you have a fairly good idea of what is coming ahead. So, coming to the bacterial cell structure. Now, this gives you the basis of the whole bacterial cell, and it could be a coccus, it could be a bacillus, but each and every one has the same structures which are going to be present, except for that the shape may vary from a coccus to a bacillus. So this is a bacterial cell. It has various structures, and we'll study each one of them. But you need to remember this diagram very well. It does not need to be such a beautiful picture, but it needs to be a picture which reflects the bacterial cell and which reflects each and every structure in its proper place. If this is reflected as a phylus, then this should look like a phylus. Or if this is a fimbriate, then it should be a fimbriate. And this is a flagellum. It should be in its proper place. You cannot put the things at their wrong places and expect that the examiner is going to give you full marks for your diagram. It should be a pencil sketch. It's not necessary. It should be a beautiful colored diagram. You don't have time to make these diagrams in this way. Just practice with a pencil and make them perfect in the sense that they reflect everything. So when we go from the outside to the inner side, we are going to see first of all the appendages, which include number one, this is the flagellum. Number two is the fimbriate. These short hair-like structures, they are the fimbriate. And number three are the phyla. Phyla is comparatively bigger than a fimbriate, but shorter than a flagellum. And all three of them, they have different functions. Then we come to the surface layers. And the surface layers include number one is the capsule or a slime layer. And it is not necessary that all organisms have a capsule. Number two, then we have the cell wall. And finally, we have the cell membrane. So these constitute the surface layers of the bacteria. Then if you go inside, we have the cytoplasm. And this cytoplasm will contain the nuclear material. It's without a nuclear membrane. It will contain the ribosomes. It will contain the inclusion bodies. It will contain a plasmid. Plasmid, again, is a small piece of DNA or RNA, which is going to be present within the cytoplasm of the bacteria. And then we have the special structures which we are going to study, that is the endospore. So first of all, we'll go through the appendage. The number one appendage which we are going to study is the flagella, which is made up of three parts: a filament, a hook, and a basal body. So this is the picture. This is the filament which you will see in a bacteria which is going outside, and it is long and it is movable. This is the hook, which is the pivot at which allows this filament to rotate counterclockwise or a clockwise direction. And then we have the rings, which are known as the basal body. And this basal body is in two rings. One is going to anchor the cell wall, the other is going to anchor the cell membrane. So these two anchor points, one in the cell membrane and one in the cell wall, makes it fixed within the cell surface layers. The hook allows it to rotate counterclockwise or clockwise, and the flagella is then going to move about, or it is going to just oscillate or go from one side to the other and help in the motility of the bacteria. Depending on the arrangement of these flagella, they can be called as polar or monotrichous when they have just one pole, one flagellum at the pole. They can be lophotrichous when they have a tuft or a number of flagella at one pole. They can be amphitrichous when they have these flagella single but at both poles. Then they are peritrichous when all these flagella, they are around the whole of the bacterial cell surface. And then we have the amphilophotrichous tuft of flagella at both ends. And the flagella is basically present in the bacillary bacterial population. You will not see the flagella in the cocci. You will only see them in the bacilli. So these are the electron microscope pictures of these organisms. Over here, this is a spiral. Again, it has the flagella at both ends, and this is amphitrichous, this is monotrichous, this is all over, and therefore this is lophotrichous, and this is all over, which is peritrichous. Lophotrichous is at both the poles. Now, how does this flagella help in the movement? When we have a peritrichous organism, that means that the flagella are all over the surface. During movement, all of these, they come to one direction. For example, in this case, they have gone towards the left, and they become a bundle. And this bundle then intertwines with each other, and it starts a counterclockwise rotation. And when it has a counterclockwise rotation, it is going to move forward. After some time, this whole bundle is going to scatter. It's going to reopen. And at this stage, it is just going to remain vibrating or tumbling at this place at which it is. So this movement is called a run, and this movement is called a tumble. Again, after some time, it is going to become a bundle, and then it will start moving again towards the particular direction. So for movement, it has a counterclockwise direction. Tumbling, it has a clockwise rotation. Similarly, if it has just even single flagellum, it's going to have the same movement, except that during the counterclockwise, it's moved forward, and with the clockwise rotation, it's going to move backward. What does this movement occur in response to? Either it can be an attractant, it could be something which is attracting the bacteria, some chemical, something from nutrition, food, which is allowing this bacteria to move towards it. And it is going to have a run and a tumble and a zigzag sort of a movement. Or if it finds something which is dangerous, then it is going to be repelled and it is going to go back. Number two are the axial filaments. Axial filaments are basically going to be present in the spirochetes or the spiral bacteria. And these are going to be present between the cell wall and you have an outer sheet. These spiral bacteria, apart from the cell wall, they have outer sheet, and between these two, you will find these filaments going around and around. And they are going to be seen basically in the organisms which are known as the spirochetes. Again, they are responsible for the Number third is the fimbriae. Fimbriae, as you can see, they are very small hair-like projections which are coming from the cell surface. And their function is basically to adhere these bacteria to different cells. For example, if we take the intestinal bacteria, How do they remain within the intestine despite the intestinal movements, the bowel movements which we are having, which forces all the material inside our body to go towards the outside? It is because of these small hair-like structures which are adhesive, which have certain chemicals or secretions attached to them, or which they are releasing, which allows them to get attached to the intestinal epithelial cells, and with the normal bowel excuse me, with the normal bowel movements, they are not going to be forced outside to the cell, outside of the intestinal lumen. Then we have the fourth structure, which is the pili. Pili, these are again protein appendages. The protein which is known is known as this is the pilus. That is known as pilin, and it belongs to the class of proteins which are called lectins, and it binds to the cell surface polysaccharide. So again, it's a binding structure. It is going to bind these organisms to the various epithelial cells, and all those organisms which have both the pilin and the fimbriae, they are going to be more adherent to the cell surfaces as compared to those which do not have these pili. And these pili, they are mostly found in the gram-negative bacteria, and as I told you, they are smaller than the flagella. One important thing which I need to tell you is that they adhere to the bacterial surfaces depending upon the various types. They are going to be of various types, and some bacteria will have them, some will not. And the antibodies which our body produces can block these and prevent an infection. For example, E. coli has certain pili which are going to allow it to remain attached to the intestinal epithelium in the bladder, and therefore they are going to produce a urinary tract infection. Some of them will be responsible for genetic information, and some of them will allow the bacteria to float in an environment and reach the surface where it can get more oxygen. So this was for today. If you have any questions, I would welcome them, and you can put them up on the WhatsApp page, and I will answer them gladly. Thank you so much for all your time and your attention. Thank you, Richard.